Hi everyone, this is a lecture on cell membranes, which are found in chapter 4.3 and page 56 through 57 of your textbook. In this, we're going to cover the cell membrane's anatomy, which means the structures in the cell membrane, and we're going to cover a little bit about what each of the structures do. Okay, so the cell membrane is also sometimes called the plasma membrane, and this just refers to the outer boundary of your cell, which separates the cell from the environment. And this cell membrane is mainly made or primarily made of a phospholipid bilayer, meaning two layers of phospholipid molecules. Now, the characteristics of these phospholipid molecules ends up being that it creates what is known as a semi-permeable membrane, also known as selectively permeable. Permeable means the ability for something to get through. So if you have a bag that's really permeable, water might flow through it. But in this case, when we use the word semi or selectively, it's saying that some things can get through, but not everything can get through easily. So in this case, what the cell is ultimately trying to do is to allow certain materials in, usually food materials or other materials it needs to do its function, and it needs to get rid of waste, and it needs products to be removed as well, because some cells specialize in making stuff to help the body or to help the colony of other fellow organisms around it. These are some examples of what you may want in and what you may want out of your cell. And you'll notice I listed water and salt on both sides because we're going to learn later on the concentration of your cell is very important, and sometimes you'll need to remove salt, sometimes you'll need to remove water or add either of those things to get the balance just right, comparing the cell to its environment, but that's going to be a whole nother chapter we'll talk about in the future. Now, for phospholipids, we want to remind ourselves of the structure of the phospholipid. So a phospholipid, just reminding you, has a phosphate head, which is ionic, and because it's ionic, it means it really, really likes water. It is hydrophilic. The other side of the phospholipid molecule consists of two fatty acid tails, which are very nonpolar. So now we have a nonpolar side of this molecule and a polar side of this molecule. And what ends up happening is when you stick a lot of phospholipids in water, it ends up creating a structure where it arranges as a bilayer so that the side that likes water, the phosphate side, ends up facing the outside, and the side that hates water ends up avoiding touching the environment around it that's full of water. So sometimes we get really lazy when drawing lots of phospholipids. So you're going to see this simplified model of a phospholipid quite a bit, where there's a circle that represents the phosphate ionic side, and then two lines to represent the fatty acid side. But the way it arranges is quite like this. And what basically is essentially happening is the inside of your cell is watery, the outside of your cell is watery, so that the phosphate ends of the phospholipid are going to face that way. And the tail sides are going to face each other because the tails on either side of the phospholipid layers don't want to interact with water. So by facing to the middle region, they get to avoid the water. But what ends up effectively occurring then is you have this wall of phospholipids with a very thick region in the middle that is hydrophobic due to those nonpolar molecules. And that's going to prevent stuff that's naturally found on the outside or the inside of the cell from crossing, because the stuff that's on the outside and inside of the cell is, generally speaking, hydrophilic. So it's going to have a harder time crossing that middle region. Some stuff can cross, though. But this is another image of kind of that arrangement of phospholipid bilayer in the cell membrane. This isn't quite a bilayer. This is called a micelle, and it's just what phospholipids make when there's not enough phospholipids around to create a, big bubble like a cell. Now, what can cross this phospholipid bilayer? Well, the phospholipids itself, they're attached through what's called interactions, hydrophobic interactions and some hydrophilic interactions. So while they are attached in a way that's pretty good and pretty stable, they also are not immobile. They're not glued together. So what that does mean is it makes it a little bit fluid, which means it lets it be flexible, but also means some stuff can slip through. But you got to remember, if it slips past the hydrophilic region, it now has to get past this entire huge hydrophobic region before it can pop through the other side. So what ends up effectively happening is molecules that can cross past the membrane very simply are things that are small and nonpolar. They don't have a problem. However, if you're polar, you're going to have a little bit harder problem. So small polar molecules are small enough to slip through and get past that nonpolar region, but it's going to take some time because it has to, get its way through this region that doesn't really like its charges, or it doesn't really like this region before it can get to the other side. However, that also means that things that are very charged have no hope of getting past that lipid layer. And in addition to that, things that are large and polar, because it's not just charged, but it's also quite large, it's going to have a harder time getting through that huge internal nonpolar hydrophobic region. Okay, so definitely write this stuff down so that you know what can cross and what can't cross due to that relationship of polar on the edges and nonpolar in that middle region. Now, you might wonder about water because water is such an essential thing that needs to get through the cell. So yes, some water can get past the membrane without assistance, but because water is polar, what typically happens is you have what's called transport proteins, and we'll get into that in a little bit, that actually assist the water to get through very quickly. We call these aquaporins. This you don't super need in your notes, but if you're ever wondering, wait, how does water get through the cell since it's so critical? You have permanent transport doorways in your cell membrane that let the water specifically get across. Okay, so the connection of the phospholipids in the membrane is important, but in addition to that, there's a lot of proteins that are associated with the membrane that help it do its job. So the combination of phospholipids and all the proteins embedded within it looks like a collage, a mosaic, one might say, and that's what we call the membrane structure, a fluid mosaic model. It is a flexible model of proteins and fats or lipids that are connected through interactions, and so it creates a mosaic connected together that's flexible, a fluid mosaic model. Also, another term you're going to hear sometimes is the ECM, or the extracellular matrix. We're not going to talk about that too much in this class. It is actually very important, though, to the structure of organs and tissues in your body, and I covered it a lot in my class in anatomy. But for here, as far as you need to know, is there is stuff on the outside of your cell that is extra to the cell that the cell does interact with, and oftentimes it's made of carbohydrates, proteins, and mixes of that are on the outside of the cell, and that sometimes help the cell connect and attach to places or do some other jobs. Okay, so some examples of the proteins that might be found in the membrane can be seen in the image in your textbook, and there's different kinds of proteins. We'll talk about transport proteins, receptor proteins, recognition proteins, and all that in the notes. This is a picture I really like. I've drawn this for my students on the whiteboard in the past. I do recommend that you possibly take a screenshot of this and maybe even draw it out. One really good way to study is to draw kind of a cartoony version of the cell membrane over and over again without looking at your notes, and then just kind of going, oh yes, this is a transport protein. It needs to be this shape. It does this. And that's one good way to review. I'll refer back to this picture after we go over what all these different things, like the recognition protein, the receptor protein, adhesion proteins, all of them do. Okay, so the first thing I'm going to highlight is these little green things that you're going to see embedded inside the membrane. So that is actually an image of what we call cholesterol. So these are steroid lipids. So we have our phospholipids, and now we have our steroid lipids. And these are steroid lipids that actually help your membranes stay the right amount of flexible. So you want your membranes to stay solid, but not too solid. You want it firm enough that it retains its shape, but you don't want it so firm that it's not able to move and it's not able to pinch off and move and help the cell do its job. So you need it firm but flexible. And one of the problems is if things are too warm, fats melt. You know that. Butter will melt if it's too hot. But if things are too cold, then it gets real solid and hard to move around and bend. So cholesterol actually is, in a way, it buffers. So it's not a buffer the way we've learned about acid-base buffer, but it helps to stabilize and keep the membrane consistency correct. Another way the membrane helps control itself is the fatty acid tails. So what actually happens is these fatty acid tails can be unsaturated or saturated. If you forgot, unsaturated means it has double bonds inside its fatty acid tail, while saturated doesn't. But one of the big things here is saturated fats like butter and stuff like that are typically pretty solid, while unsaturated fats are typically at room temperature pretty liquid. So if you're in a cold environment, what actually happens is within the cell, you can see it inside animal cells that live in cold environments, is their phospholipids themselves actually become a little bit more unsaturated. While if you're in a very hot environment, organisms that live in very hot environments, typically their phospholipid tails will actually be saturated or more saturated than otherwise. So there's two ways we can get our membranes to be the right flexibility. Cholesterol can go either way. It helps it from going too far in one direction or the other. Or we can manipulate the phospholipids itself, but this one is hard to shift one way or another quickly, and it happens over time in an organism that lives for a long time in an environment that might be very cold and needs more flexibility, or an environment that might be very hot and needs less flexibility. Okay, so now let's get into all the proteins that you see inside the membrane. So there's two kinds of protein directions, or I guess locations. So we call them peripheral proteins and integral proteins. So integral proteins literally punch through the membrane. They go all the way through the membrane, or at least deep into the membrane. Peripheral proteins pretty much just kind of sit on the edges. I'm kind of drawing a different color. They sort of sit on the edges of the membrane. They sit on the peripherals, the sides of the membrane, but they don't go into that very nonpolar region. Okay? So some proteins have to be integral. They must go through the membrane. And some proteins could be either integral or peripheral. Either of those are okay. Now, what allows a protein to punch through the membrane is, remember, or proteins could be polar or nonpolar depending on the special R groups. So if you want a protein to sit inside the membrane, what you're going to do is have a middle region of that protein be nonpolar, so it can sit where the nonpolar fatty acid tails are, and then the outer edges be polar, so they can sit comfortably in the phosphate region. And you can see that here. So we have polar regions and nonpolar regions, which allow this protein, which is what's known as integral protein, the ones that go all the way through, the ones that are integrated, to go through the membrane. All right? Peripheral proteins will have a lot more polar stuff than nonpolar because they're sitting just on the edges. They don't have to go through. Okay, so now let's classify proteins by their function. So if we classify our proteins by their location, we can say peripheral or integral. If we want to classify it by what they do, then these are different things they may do. They may include enzyme activity. They may work as a receptor. They may work as what's called an identity marker. These cell identity markers we also call recognition proteins, and we'll go over these. And they may sometimes be called transporter proteins, also known as transport proteins. And they can also be involved in attachment to other materials or attachment to other cells. And we're going to go over each of these functions. So, pretty much, transport proteins have to go all the way through. The whole point of a transport protein is to deliver something from one side of the cell to the other side of the cell. You can't get from one side of the cell to the other side of the cell, sorry about that, without a protein that punches all the way through. Okay? Now, some other things that your proteins can do is they can be integral or peripheral. They can be adhesion proteins. In this case, adhesion proteins attach the cell to another cell or to another surface. And they can be receptor proteins, which means that they bind to materials either inside the cell or outside the cell, and that causes a change to the cell membrane or the cell itself. So you can kind of see that here. This protein itself is going to bind to this molecule, and the binding of the molecule will cause a change, a reaction, an enzymatic shift on the other side of the cell. So it gets the signal on one side, it does sort of a relay race to the other side of the cell while something happens. And then this example of adhesion proteins, where that protein assists gluing that cell or cell membrane to something. Some other membrane proteins that can be either integral or peripheral, and reminding you, integral means it punches through or goes through a lot of the membrane, and peripheral means it stays kind of towards the edges of the membrane. And just an example, in this image, all the proteins are integral, other than a couple, like this protein is not integral, but all the other proteins are integral. So just a reminder that integral is just location, peripheral is just location. But two other examples is enzymes. So enzymes, we've talked about them they a little bit, but we haven't really detailed them yet. And they basically are proteins that help chemical reactions occur quickly or more quickly in the cell. Like they help make it easier or harder, or not make it easier, help it make it easier to either make or break a bond. There are also something called recognition proteins. So recognition proteins are also known as surface ID markers. I consider them like ID badges. So what happens is you'll have a protein that has some kind of weird sugar carbohydrate shape on the edge of the protein, and this actually functions as an ID tag, such as used in the immune system. And what happens is it helps your body identify what cells are theirs versus what cells are not theirs. So if your immune system recognize, oh, the ID badge is wrong, they'll murder that cell. So that helps prevent bacteria from growing in unwanted places in your body. Or if a virus infects your cell, then some of the surface identifier markers might start looking weird, and those weird markers will flag and let the body know, oh, murder this cell. Because it's not doing what it should do. Okay, now just remember, these are all potential functions of membrane proteins. So what actually does happen in reality, because these are just models, is in reality the actual way it works in your cells is more complicated than that. And what happens is many membrane proteins could do two things. All right, you might have something that is both an enzyme and a receptor protein, because like this one, you can kind of see it be both, because if binding that molecule makes this protein recreate an actual reaction that involves the breaking or making of a bond, then it's also an enzyme. So it's both a receptor and an enzyme. Some transporters won't work or open up their doorway unless they bind to the right molecule to open the door. And that would be an example of both a receptor and a transport protein. So you get things that can do more than one. All right. You might also have two different proteins working together. So in this instance, you might have a receptor protein, right? The one that binds to things to help facilitate some kind of change or response in the cell. And that change in response might be the opening of a transport protein, or it might be the creation of a reaction that breaks a bond in an enzyme. Okay? So, just realize, even though we're learning these things in isolation, the actual reality of what's occurring in your cell is a little bit more complicated beyond that. Okay, another thing I wanted to mention is I noted that there was some proteins that are attached to sugars, in particular, to help with recognition, to help work as an ID badge. And a lot of these things are very critical to the immune system. So just be aware of that. So some recognition proteins have a very important role in the cell, and it helps a lot with cell communication. So a lot of cells put a lot of sugars on the outside of their cells to help it interact with the immune system or other cells in the body. So the way they do it is by just taking that sugar and, merging it with the protein that is naturally found in the membrane, or with the phospholipids that are naturally found in the membrane. So when you take a sugar and covalently bond it to a protein, we call that a glycoprotein. And when we take a sugar and we covalently bond it to a lipid, like this guy over here, we call it a glycolipid. But all of these are involved in some kind of cell recognition, communication thing going on. Now, I didn't make official notes on this, but I did want to mention that some of these proteins are actually really critical to what's happening in the world right now. So you might have heard of ACE2 receptor. So you'll notice here we talked about receptor proteins, which are proteins that can initiate a response. Sometimes that response is creating transportation of something from the outside of the cell to the inside of the cell, or taking stuff and causing the inside of the cell to release things to the outside. And the ACE2 receptor, for those of you who have heard about it, is a receptor that is found on human cells that allows for the intake of certain material. And unfortunately, the virus SARS-CoV-2, also known as the virus that causes COVID-19, it actually binds to the receptor protein that is in our cells to encourage those cells that have the receptor protein to swallow up the virus and allow the virus to get access to the inside of the cell. And unfortunately for us, the locations of the cells in our body that have the receptor protein that allow the cell to go, ooh, this is something I need, and swallow the virus, those cells are located in your lungs. They're located in the back of your throat, and that's for initial infection. But what creates the really scary side effects is it turns out a lot of your blood vessels, which are found everywhere in your body, including your kidneys and your brain and your heart and your skin, those all have lots of ACE2 receptors. And unfortunately for some of us, we might already have pre-existing blood vessel damage, which is some of the people that are in high-risk groups, and some people genetically have a higher expression of these receptor proteins, which also makes them more vulnerable to the virus. But in those people's cases, because their health isn't already have any prior damage, they just happen to be overexpressing this receptor that really likes to take in the virus, they don't find out if they have it until they get a really severe infection. So most people don't have that instance, but that accounts for some people who don't have prior existing preconditions that end up having a more severe reaction to the virus than others. And that's down to that receptor protein, the very famous receptor protein now called the ACE2 receptor protein. And why do we have that in our bodies? Because its major job is to regulate your blood pressure. Okay, so that's not in your notes, but I think it does give you some context on what's been going on in the world. But basically, our main takeaways in terms of the material you should know for this unit is that cell membranes are very critical for allowing a cell to be specific to serving the needs of the organisms. And the phospholipid bilayer allows that the cell membrane itself to be selective because it has an internal hydrophobic barrier that prevents a lot of materials from crossing that are naturally found on the inside of the cell and the outside of the cell. And the outside and inside of the cell is touching the hydrophilic side of the phospholipids that are making that bilayer. And because proteins can be hydrophobic and hydrophilic, they're allowed to be part of the mosaic, the fluid mosaic membrane, I should say, that is so critical to allowing the cell to do reactions and to facilitate transport of stuff that can't naturally cross the bilayer. And a big one there is transport proteins, which really facilitate the crossing across the membrane, is called an integral protein, which is a location of a protein that goes all the way through the membrane, versus the other types of proteins we reviewed, like enzymes, like recognition proteins, like receptor proteins, that could be either integral or peripheral on the edges. But all of these proteins play a really critical role in what the cell can do and what the cell can respond to. And on that note, please notice all the vocabulary that was here that was new to the lecture, and make sure you understand these words. And beyond this, you should also know what allows the cell membrane to be more fluid or more solid, and also what material can pass through the membrane easily versus what can't. All right, good luck, and move on to any other work you have, and I will talk to you in the next lecture. Bye.